will be arriving. This meeting's being recorded, and I know we're going to have a really exciting discussion today, and I'm looking forward to hearing from our speakers and panelists and learning a lot from them. So my name is John Scott. I'm the Director of the Division of Biostatistics in FDA's Center for Biologics Evaluation and Research. And I'm going to take just a few minutes to sort of give some background and introduce what's going on today. But really, the speakers and the panelists are the stars of the show today. So, starting with sort of the obvious, we rely so much on clinical trials to support our critical regulatory and public health decisions. They form the backbone of evidence of safety and effectiveness needed for drug and biologic approval. I think most people know that the costs and complexity of trials have ballooned in recent decades. Some of the numbers people say are somewhat eye-watering in terms of cost. And complexity, the questions we're trying to answer just get more and more complicated as science progresses. So consequently, there's really been a need for innovative approaches to answer complex questions and to improve trial efficiency. And some of those approaches include various adaptive designs, Bayesian approaches, and potentially incorporating external data in trial analysis and design. And one result of this need was FDA's complex innovative trial design, which for reasons I won't belabor, we don't have a T in the abbreviation for, we call it CID review program. A bit of the history of the CID review program. Under PDUFA VI, the sixth authorization of the Prescription Drug User Fee Act, which ran from 2017 to 2022, Congress gave FDA a mandate to facilitate the advancement and use of complex innovative designs in regulatory decision-making. And there were several sort of subparts of that commitment, which included developing staff capacity, convening a public workshop. We had a workshop much like this one 5 years ago, publishing draft guidance, and developing sort of review procedures and templates as appropriate. But importantly, the kind of main thing was the requirement for FDA to conduct a pilot program for the review of CID proposals. So what that pilot program was, it's a joint effort between FDA's Center for Drugs, thanks, I thought it was a little high, between FDA's Center for Drugs and Center for Biologics Evaluation and Research. What happens is sponsors submit designs to be considered by FDA, and if the designs are accepted into the program, they have the opportunity to engage with the regulatory review team on those designs via 2 additional meetings, typically focused specifically on the more technical aspects of the designs. FDA will select up to 2 of these submissions per quarter, and then one of the unique features of this program is that we form an agreement with the applicant on elements of the proposal that can be publicly disclosed so that we can use the designs as case studies for others for outreach and education. Those CID meetings are led by the biostatistics groups in FDA. So in CDER, it's the Office of Biostatistics. In CBER, the Division of Biostatistics. But there's, of course, participation from clinical teams and from all relevant disciplines. So again, this ran from 2017 to 2022. Over that time, we accepted 6 submissions across several therapeutic areas, including neurology, analgesia, rheumatology, oncology, and several of them were in adult or pediatric rare diseases. And the methodologies or designs that were used included Bayesian hierarchical models, the use of informative priors to bring in structure and information, and master protocol designs. I mentioned that PDUFA VI committed us to publish guidance on CID. We broke that up into 2 different guidances. One, a guidance on adaptive designs for clinical trials for drugs and biologics, and the other, a guidance on interacting with the FDA on CID proposals. So that brings us to PDUFA VII, 7th iteration of the Prescription Drug User Fee Act, which we're in now. It runs from 2022 to 2027. The objective is similar to PDUFA VI. says to facilitate the advancement and use of complex adaptive, Bayesian, and other novel clinical trial designs. One interesting difference is that the objective now explicitly mentions Bayesian. And we have our sort of goals under this, which include continuing to develop staff capacity for CID review, to continue the paired meeting program, which we no longer call a pilot, we just call it the paired meeting program, to convene the public workshop that we're having today, and to publish a draft guidance on the use of Bayesian methods in drug and biologic trials. So in today's workshop, our goals are to discuss aspects of complex adaptive, Bayesian, and other novel clinical trial designs. And specific topics we were asked to consider include considerations for external data sources, Bayesian statistical methods, simulations, that is, trial simulations for operating characteristics, and issues with clinical trial implementation. And the way we're structuring today, I hope everybody has the agenda for specifics, but we're going to start in the morning with 3 case studies of successful CID proposals, followed by a panel discussion, including the speakers and additional panelists. I'm not going to go through people's biographies in detail, but we have a panel today of extremely distinguished speakers and panelists. The detailed biographies are posted on our event webpage, so I'm just going to introduce people quite briefly now. Let's see, I think it is in order from my immediate left. Frank Bretz is a distinguished quantitative research scientist at Novartis. Dean Folman is chief of the Biostatistics Research Branch at the National Institute of Allergy and Infectious Disease at NIH. Frank Harrell, who is joining us remotely, is professor of biostatistics at Vanderbilt University School of Medicine and also an expert biostatistics advisor to FDA's Center for Drugs Evaluation and Research. Rebecca Hubbard is professor of biostatistics at the University of Pennsylvania. Jack Lee is professor of biostatistics and Kennedy Foundation Chair in Cancer Research at MD Anderson Cancer Center. Roger Lewis is senior physician in the Los Angeles County Department of Health Services, professor of emergency medicine at the David Geffen School of Medicine at UCLA, and a senior medical scientist at Berry Consultants. Herb Pang is expert statistical scientist in PD data sciences at Genentech Roche, and Karen Price is vice president, Statistical Innovation Center, Advanced Analytics at Eli Lilly. Oh, and I somehow missed Steve Ruberg. Steve Ruberg is a former employee of Eli Lilly and is currently the principal of Analytics Consulting. Is that correct? Analytics Thinking. I'm so sorry for the neglect, Steve. That doesn't reflect your importance to the panel. Okay. So before we get started, I wanted to thank, there were a lot of people involved in making this workshop come together, but in particular and especially I wanted to thank Tuan Pham, who is the CID project coordinator for CEDAR. Tuan was instrumental in every aspect of this, ranging from speaker contact to agenda formation to the Federal Register notice to coordinating catering, and we wouldn't be here without him today. Christopher Egelebo is the CID project coordinator for CBER and also participated quite a lot in organizing today's workshop. We have a lot of internal folks at FDA who work on CID and who contributed in some way to this effort. There's a steering committee, a proposal selection committee, and an education subcommittee. And of course, when these proposals come in, there are CEDAR and CBER reviewers who review them. And then finally, the White Oak AV team, who I'm grateful that they turned down my mic and are doing many other things behind the scenes. And then finally, before we begin, I really wanted to give a very special thanks and recognition to my colleague, Dr. Dion Price, who tragically passed away 2 weeks ago, but otherwise would be the one here giving this introduction. Many of you in the audience knew Dion or were touched by her leadership in some way, but for those who weren't fortunate enough to know her, Dion was Deputy Director of the Office of Biostatistics in FDA CEDAR and past president of the American Statistical Association. Absolutely nobody had a more significant impact on FDA's CID review program and how we treat complex designs than Dion. She led the program for CEDAR, starting with PDUFA VI and through the PDUFA VII reauthorization, and she in particular was instrumental in the PDUFA VII negotiation process that led to the continuation of the CID program. Her influence on every major decision for the program and most minor decisions for the program couldn't be overstated, ranging from process implementation, proposal screening and selection, and external communication and outreach. And CID was just one of many things she did. Dion brought outstanding judgment, strong leadership, and really, truly unparalleled diplomacy to everything she did, and kindness. And we miss her a lot. She would want us to have the workshop, I'm sure. So we'll proceed. And our first speaker is Dr. Roger Lewis, who's going to be speaking about the CHIPS trial, which was an adaptive storage duration finding trial for platelets. And this trial is, of the three, this is one that wasn't a formal proposal to the CID meeting program. It just came through the ordinary IND review path, which illustrates that CIDs aren't restricted only to the program. I wanted to mention that if people have questions about the talks specifically, if you're on Zoom, you can put them in the Q&A and we may be able to get to them later, but we will also have a specific designated time later for audience questions, both in person and by Zoom. Okay, Roger, thank you.
Great. Thank you, John. It's a pleasure to be here. I'd like to thank the organizers for this opportunity. And as John mentioned, this is not a trial that was designed through the CID program, but I think that the positive interactions with the agency throughout the development of the trial illustrate the influence of the CID program on the agency's thinking. These are my disclosures. Most importantly, relevant to this, I'm the senior medical scientist at Berry Consultants and also an inactive special government employee. So the CHIPS trial, first of all, I need to point out that I'm not the person doing the hard work of the trial. It's led by multiple principal investigators. There's been a very large statistical design team involved in both the design and implementation, a data coordinating center at the University of Utah, and it is funded by the U.S. Army Medical Research and Development Command through the U.S. Army Medical Research Acquisition Activity. So the background is that in the United States, platelets, a critically important component of our clotting mechanism, are collected and then stored at room temperature. And because of the potential for a small amount of bacterial contamination, the storage at room temperature means they can only be stored for a short period of time, typically 5 days, before they must either be used or discarded. There are some provisions for extending that storage time up to 7 days if you test the units individually for contamination. But because of this short period of storage, many hospitals with relatively low volumes or rates of blood product usage are unable to maintain platelets. In fact, 10% of the hospitals that acquire red blood cells through the American Red Cross actually don't even attempt to acquire platelets. That lack of availability of platelets put patients with bleeding, either due to platelet inadequacy or, for example, due to a major trauma, at tremendous risk for adverse outcomes. If we were able to store platelets for a longer period, that would substantially mitigate the challenges associated with maintaining platelet availability in a variety of areas, including austere settings. So the objective of the CHIPS trial is to demonstrate that platelets that are stored at 4 degrees centigrade, what we'll call cold stored platelets, are non-inferior or potentially even superior in terms of their ability to treat active bleeding, so-called hemostatic efficacy, when compared to standard room temperature platelets stored at 22 degrees. And I may refer to that as either room temperature platelets or warm platelets. And we're going to evaluate that in adult and pediatric patients who are requiring complex surgery and who are actively bleeding as a result of that surgery. This is not because that patient population has any particular bleeding mechanism, but this is a well-characterized setting in which to evaluate hemostatic efficacy. The secondary objective is to determine the maximum storage time up to a potential of 21 days over which the product maintains non-inferiority when compared to room temperature stored platelets. So I want to just take a second and circle back to why we use adaptive approaches in many settings in which we have sparse information related to key components of a clinical trial design. So I think many of us have had the experience that when we are designing a clinical trial, we almost never have enough information in order to know the optimal design. And specifically in this case, we don't know how long you can store platelets over a relatively wide range of uncertainty, say 7 to 21 days. So we don't know what is the storage duration over which we ought to be exploring the hemostatic efficacy. But once patients are enrolled in a clinical trial, data start to accumulate that reduce that uncertainty that existed at the time of the design of the trial. An adaptive trial is designed to take advantage of that stream of initially sparse but increasing information in order to make changes according to pre-specified rules and to mitigate some of the risks that were associated with the initial uncertainty. In certain cases, that can increase the probability of getting the right answer at the end of the trial or improve the trial efficiency. So specifically in this case, we're going to be using incoming data to help us know where we should be focusing our attention on the continuum of the storage duration of platelets. So an adaptive trial can be put into this generic framework where in the upper left we begin with an initial set of sampling rules. So for example, we might start with a particular storage duration or a particular randomization ratio. We take a first look at the data and analyze the data, ask if there's a good reason to stop the trial, and if not, we may revise randomization, sampling, or other rules in response to the partial accumulated information at the time of that interim analysis. We then continue with additional data collection according to those new rules. That process can continue in a circular fashion until we reach a reason for stopping the trial, for example, being able to draw a firm conclusion regarding efficacy or inferiority or reaching the maximum sample size planned for the trial. So we're going to place the CHIPS trial design into this framework. Now, the CHIPS design itself is a fixed randomization trial with 2-to-1 randomization of cold stored platelets to room temperature platelets in order to increase the both experience and safety database associated with cold stored platelets, and because we need to explore the storage duration relationship to hemostatic efficacy. The primary endpoint is a 5-point bleeding score, a hemostatic efficacy score, and importantly, lower scores are better. 1 is good, 5 is bad. The two arms are treated differently because we consider room temperature platelets to be a homogeneous treatment. Even though platelets can be stored from 0 to 5 days typically, the processing time means that most room temperature platelets are 3 to 5 days old at the time of transfusion, so we consider those to be a single treatment. In contrast, when you receive cold stored platelets as a patient, you typically receive a set of platelets that will have a storage duration, and you may have another number of units of platelets that have a different storage duration. So we're going to characterize the treatment of a patient by the weighted mean storage duration of the administered cold stored platelets. So in the room temperature arm, platelets don't have an age, but in the cold stored arm, they do have an age that's defined by this average of the age of the platelets administered. It's a non-inferiority trial because the advantages in terms of the ability to store the platelets mean that this would be an important part of our options for treating these patients, even if they were not quite as effective as room temperature platelets, and the non-inferiority margin is 1 unit on the bleeding score, and we're going to demonstrate type 1 error control through simulation. We're going to have some adaptive rules for changing the maximum storage duration of cold stored platelets as the trial progresses, and the trial is designed with a fixed maximum sample size with 1,000 patients, 200 and interim analyses after every 200 patients. That's the overall structure. The underlying inferential model assumes that the hemostatic efficacy score, the mean hemostatic efficacy score for cold stored platelets, is a function of the storage duration X, whereas I said earlier, a smaller score is better. We'll let mu sub warm be the true mean hemostatic efficacy score for room temperature platelets, which is a single number. It's not dependent on the age of the warm stored platelets. The efficacy of the cold stored platelets is modeled as a monotonic piecewise linear regression model. The monotonic there I call the "this is not wine" assumption. We assume that platelets do not get better with age. So it assumes that as the platelets are stored longer, their hemostatic efficacy will remain the same or it will decrease. The null hypothesis is that there is no storage duration for which the hemostatic effect of cold stored platelets is within 1 unit of that of warm stored platelets. And the alternative hypothesis is that there is some storage duration of 7 days or greater for which the hemostatic efficacy of the cold stored platelets is non-inferior to warm stored platelets. At the end of the trial, if it does not stop for futility, so after 1,000 patients, we look for the longest duration of storage for which the model-based prediction is that there is a 97.5% posterior probability that the cold stored platelets are non-inferior to warm. If that is met, then there is a gated superiority hypothesis that has a more stringent criteria of 98.3% posterior probability, and it also has a requirement for super superiority that's denoted by the little delta of X, which is required to maintain type 1 error control for the superiority hypothesis because of the monotonic assumption that's built into the model. Again, that parameter is determined through simulation. At each interim analysis, we want to have an opportunity to alter the maximum cold stored duration. So at each interim analysis, we ask, based on the current model for the relationship between storage duration and hemostatic efficacy, what is the longest duration for which we predict there is at least a 33% or 1/3 chance that that storage duration is truly non-inferior. And we consider that a candidate for a new maximum duration of storage. If that candidate is less than 7 days and the probability of non-inferiority at 7 days is less than 10%, the trial stops for futility. So that says if we can't store platelets for at least 7 days in the cold and have any reasonable chance they are non-inferior, then we stop the trial. But if the candidate duration of maximum storage is greater or equal to 7 days, then we take the minimum of 3 possibilities for the new storage duration for the next 200 patients. Either the candidate time of storage itself, the maximum duration plus 5 days, or 21 days. What these rules mean is that for whatever the current maximum storage duration is, we can only increase by up to 5 days, we cannot go over 21 days, and we cannot go past a storage duration for which there isn't at least a 1/3 probability of non-inferiority. There is no early stopping for success in this trial design. So I want to take a second to show you what some simulated data might look like, because this will be important for understanding how the trial plays out. So on the left side of the graph, you see a pink dot around 2. That is the mean observed hemostatic effect of the warm stored platelets. And there's some uncertainty around that estimate, and you can see the faint dots for the number of participants whose hemostatic efficacy has been the integral values 1, 2, 3, 4, and 5. If you take the observed hemostatic efficacy of the warm stored platelets and you add 1, you get the non-inferiority margin, which is shown by the horizontal yellowish line. And then for the cold stored platelets in the simulated data, you see that there's data out to approximately 17, 18 days of storage. And there is mean hemostatic efficacy scores that have been observed from the data, shown by the orange dots of various size related to the number of platelets at those various time points, and a fitted line based on this monotonically increasing model. The goal of the design is to identify where that fitted line crosses the non-inferiority margin, which will reflect the maximum storage duration for which the cold stored platelets maintain non-inferiority. So going back to the overall structure of an adaptive trial, we're going to start with a maximum cold storage duration of 7 days, the same duration that's allowed for warm stored platelets if they are tested for their sterility during their storage period. We're going to start with a first interim analysis after 200 participants. We're going to fit this model for the hemostatic efficacy of the cold stored platelets as a function of the duration of storage. We're going to make sure that the 7-day storage duration has at least a 10% probability of non-inferiority. That's the futility rule. And then we're going to apply those rules at each interim analysis to revise the maximum storage duration with the hope that over time it will gradually increase, as in a duration finding experiment, we find the maximum storage duration that maintains non-inferiority. Once we get to 1,000, then we will find the longest storage duration in which the probability of non-inferiority is greater than 97.5, and that will be the primary result of the trial. And if that is positive, we will also evaluate for superiority against the more stringent posterior probability cutoff. So here's an example trial based on simulated data to show how this plays out. I want to take a second to orient you to this graph. So in the upper left panel, the structure is the same as was in the simulated data I showed earlier. But at the start of the trial, this is after the first 200 subjects, we've only been allowing the cold stored platelets to be stored up to 7 days. So we only have data to support the model fit out to 7 days. Because of that, you can see there's tremendous uncertainty if we try to extrapolate those sparse data out to longer storage duration times. In the lower left, you see the number of subjects that have been enrolled at each interim. I'm sorry, the range of cold storage duration that has been that is allowed up to that point. In the middle of the bottom of the slide, you see the fitted probability of non-inferiority based on the model. And if you look at the right-hand column in the table in the middle bottom of the slide, you can see that for all of the durations, the model says that the posterior probability of non-inferiority is greater than 1/3. So the model would say that it would be acceptable to have your X candidate up to 21 days. But our rule for increasing the storage duration is that at no point can the number of days of storage increase by more than 5 days. So the result of this interim analysis would be to, for the next 200 patients, have the maximum length of duration be 12 days, which is the 7 days that we currently have data for, plus the 5 days, which is the maximum step forward that we are allowed to take. The bottom right-hand panel shows you the storage durations that have a probability of non-inferiority greater than the 33%, shown by the horizontal red line. And then the upper right panel shows you the sample size in the various categories, be either warm storage duration or cold storage duration in various bins. So if we let this trial go on to the next 400 subjects, the 400 subjects, you see that the current max cold storage duration was 12. We now have data in the interval from 7 days to 12 days, as well as additional data up to 7 days of cold storage. That decreases the uncertainty. Again, the models identifies all storage durations as potentially being non-inferior with a probability greater than 0.3. But then because we're at 12 days, the maximum we're allowed to move to as the storage duration is 12 plus 5 or 17. We enroll an additional 200 patients for a total of 600. And now if you look at the middle of the bottom of the slide in the table, you can see that at 17 days of storage, there's a 45% probability of non-inferiority, but by 18 days it drops below a third. So we were studying platelets out to 17 days. The model says you cannot increase the maximum storage duration because 18 days has less than a 1/3 probability of being non-inferior. So we're going to continue with the maximum of 17 days of storage, collect more data. Here's after an additional 200 patients. The model chosen duration at this point is 20 days because that's where the predicted probability of non-inferiority falls less than 0.33. And then at the end of the trial, these would be the final results. Now, it's important to note that the precision in the estimate of the efficacy at the longest storage durations is highly dependent on the support for the model out at the right-hand side, and that's going to be related to a comment that we received from the FDA during the IND review process. Okay. So again, the primary trial analysis after 1,000 patients is that either it's a negative trial because there's no cold storage duration of 7 days or longer that is non-inferior, or we identify a period of storage between 7 and 21 days inclusive that is non-inferior, and then we can also evaluate for superiority. So what are the operating characteristics of this design? In order to evaluate the design, we take the design and we make lots of different assumptions regarding the true underlying efficacy of cold stored platelets as a function of storage duration, run thousands of trials, and simply count up the trials that get an answer that is consistent with the underlying assumed truth or inconsistent. And I just want to point out that this trial can get wrong answers in a number of different ways. It can fail to identify a storage duration that exists. It can it can identify a maximum storage duration that is incorrect, meaning the platelets would actually be inferior. And it can have varying degrees of accuracy in identifying the maximum storage duration that maintains non-inferiority. So in order to evaluate type 1 error control, one has to come up with a variety of scenarios regarding the possible true relationship against which one wants to evaluate the performance of the trial. So here you see 7 different assumed relationships. The horizontal dashed line is the assumed hemostatic efficacy of warm platelets. That's at 2. The non-inferiority margin, therefore, is at 3. The broad gray line are the different assumed relationships between maximum cold storage duration and the hemostatic efficacy of the cold stored platelets. Note that every one of the gray lines goes through the point at 7 days and 3. That's the definition of the null hypothesis. And you can picture that the more gradual the slope, the more difficulty the trial is going to have identifying the correct storage duration because there's many days that are close to the non-inferiority margin. Okay. So this graph captures both the rate at which type 1 errors are made and the accuracy in identifying the maximum non-inferior storage duration. So the vertical axis is an expansion of the distance between the efficacy of the warm stored platelets and the non-inferiority margin, which is a value of 1 greater. The solid red lines are the assumed relationship between storage duration and hemostatic efficacy for cold stored platelets. The vertical dashed line is the maximum storage duration that maintains non-inferiority. The histograms, the bluish histograms, show the relative frequency with which simulated trials identify a maximum storage duration of the different numbers of days. And as long as those fall below 7, not including 7, those are negative trial results. The type 1 errors are any case in which a blue histogram falls on 7 or greater, and as I'll show at the end, it's controlled at a 0.025 level. The futility rule is based on this calculation of the posterior probability of non-inferiority at 7 days falling below 10%. And under the particular null hypothesis that's shown in the lower left, the futility rule stops the trial about half of the time. To evaluate power, one also needs a set of alternative hypotheses. So in each one of these cases, you may note that the gray broad line that represents the assumed efficacy of the cold stored platelets falls below, therefore, better hemostatic efficacy than the non-inferiority margin at 7 days. The most difficult in identifying the correct storage duration will be the more gradually sloping curves, and we'll be able to see that. This is the same presentation of the performance of the trial for those alternative hypotheses. So starting at the upper left, that's the situation in which warm stored and cold stored platelets have exactly the same efficacy, and there's in fact no decay in that efficacy for cold stored platelets over time. And you can see that the trial is very efficient in correctly identifying that the maximum storage duration of 21 days is the correct storage duration. For the case in which there is a linear relationship between storage duration and efficacy, and it crosses at 16 days, you can see that the trial identifies multiple different possible maximum storage durations in the different simulated trials, ranging from about 9 days up to 15 days, and does not overestimate with any appreciable frequency the maximum storage duration. The model is specifically structured to avoid what we call overcrossing, meaning identifying a storage duration that is too long and that would therefore put patients at risk for receiving an inferior product from a hemostatic efficacy point of view. You can see for each of the subsequent different alternative hypotheses, there's similar behavior where the trial systematically is conservative in estimating the maximum storage duration, and there are broader distributions for those curves in which there's more gradual crossing of the non-inferiority margin and a more tightly clustered set of findings across simulated trials when there is a steeper relationship between storage duration and hemostatic efficacy. So if one simulates the trial thousands of times and averages this, the behavior, this is the operating characteristics table. And I just want to point out that it's not just a question of type 1 error or power. So in the first column, you have a traditional power, and you can see that at the bottom, across those different potential shapes of the null relationship, the type 1 error is controlled at less than 0.025. For the alternative hypotheses, there is excellent power for detecting the fact that there is a storage duration greater than 7 days for which the hemostatic efficacy is non-inferior to warm stored platelets. The second column, inferior patients, tells you the number of patients out of 1,000 who received platelets that in fact were inferior because they had been stored longer than the place at which the two curves cross. And obviously we want to keep that number to a minimum because that reflects potential risk to participants in the study. The third column is the number of times the fraction of simulated trials that gets the storage duration within 3 days, which is the correct number or 1 or 2 less. And overcrossing is the frequency with which the final result for the trial includes a day of storage, even one, for which the platelets would be inferior to the warm stored platelets. That's obviously something we wanted to avoid. So I just want to point out that the original letter on the IND in which we removed the clinical hold had a specific recommendation that we try very hard to maximize the number of subjects exposed to platelets at the longest cold storage duration in order to maintain the data support for the model out at that location in storage duration. And I just want to point out that the team works incredibly hard to manage the platelet inventory in order to maximize the exposure of the longer cold stored platelets, and that is quite a challenging thing. The trial has enrolled incredibly well. I think a lot of people would be jealous of the relationship between the actual enrollment and the planned enrollment, and there's a good distribution of ages in the patient population, which should help in interpretability of the results. The trial has just recently crossed 600 patients, and therefore will be conducting its third planned interim analysis. And I'd like to finish by making a comment about an external event led by the regulatory agency that caused us to change the way the trial was being conducted. So in June of 2023, the FDA released a guidance that essentially made it acceptable to store platelets for up to 14 days when conventional platelets are not available or their use is not practical. And the language is very specific, but it essentially says that up to 14 days is okay. At that time, the trial, if it had been increasing its storage duration as designed, would have had a maximum storage duration of 7 to 12 days. It would have been somewhere in that range. So the FDA guidance immediately set an upper limit on storage duration greater than the storage duration that the trial could have gotten to. I did appreciate the guidance specifically mentions this trial, and I took this as an endorsement of the importance of completing the trial as planned. So in response to that, the DSMB for the trial reviewed a request from the investigator team to immediately increase the maximum storage duration up to 14 days, even though the design could only have gotten to 12 days at that point if everything had been accelerating or progressing as rapidly as possible. And the DSMB approved that request. This did not leak efficacy information because that decision was based on external information not related to the trial. Whereas if the investigator team had been told the maximum storage duration based on the algorithm within the trial, that would have leaked efficacy information. So I'd like to stop there. There's a number of references that give the original design and talk about the trial. And again, I'd like to thank the agency for their support in getting this trial going, and I look forward to its results. Thanks, John.
Thanks, Dr. Lewis. Please don't leave yet. So I think it's a very interesting design. I wanted to turn to the panelists and see if anybody had any questions or comments before turning to others. Dr. Pullman.
Yeah, thanks. I thought it was a really nice design. It reminded me a little bit of some studies in infectious diseases where you might randomize people when to start ART, and so there's this duration question, and also studies that look at the duration of TB therapy where you want to find the sweet spot, and you're sort of pushing the envelope on what the duration is. So I had a couple points. One is, you know, a lot of times when we're doing non-inferiority trials, we struggle with the margin, and I'd be curious about how you got the margin of 1. And the other comment is that you don't randomize to the duration, of course, and so there's, I guess, a potential for a bias to creep in where if, as time goes by, you get slow bleeders later in the study compared to early, and so the slow bleeders get the longer durations, and then you're comparing them to the lumped control group, which has slow and fast bleeders. So I just wondered if, you know, there was thought about the concern of a secular trend is sort of, confounded with longer durations in the duration arm, in the cold storage arm. But overall, I was just really impressed with it. I thought it was soup to nuts, really great.
Great. Thank you very much. So the model in which the cold platelets are being evaluated of cardiothoracic surgery has a variety of different bleeding endpoints that can be used. Chest tube output is a standard one. This is a clinical bleeding score that reflects both quantitative measures such as chest tube output and also the surgeon's experience with the surgical bed during the operation and bleeding during the next 24 hours. I think that over the duration of time that this trial is being conducted, the likelihood of significant secular trends affecting sort of the clinical impression of the bleeding score is unlikely, certainly not impossible. To my knowledge, there have not been any substantial changes in clinical care practice or the use of ancillary treatments of bleeding during the trial. With respect to the non-inferiority margin, because the goal here is to improve the availability of platelets in both rural hospitals and potentially other austere settings, the non-inferiority margin is really a value judgment related to what is the minimum efficacy of a product that would make it a useful adjunct to controlling bleeding in these settings in which no platelets are currently available. And so it's a value judgment of the investigator and blood banking team.
Yeah, Roger, yeah, I think this is an excellent example illustrate the complexity and the adaptivity needed, for a trial like this, like in the CID setting. Okay? And thank you for the very comprehensive and thoughtful, design. My general question is that we can think about CID is a very complex machine with many knobs you can adjust, right? So you like to have the output is also complex. It's not just one-dimensional output, right? So, for example, you like to say if a noodle machine make noodles, right? And then there are many things input and many knobs to adjust, and the output, there are also many measurements of the output. So my question is that during this, so of course you have certain essential design characteristics you like to achieve, like control type 1 error rate and reach certain power. But beyond that, are there some criteria that you will look for to quote unquote optimize the trial? And again, in a very simple setting, there are many optimal design available. But in complex setting like this, maybe there's no one single criteria that you try to optimize. Okay, but there are many knobs you can turn, right? For example, how many interim analysis when you do the interim analysis, right? And then what's the probability confidence level, for futility and for declared efficacy, right? And randomization ratio, right? So there are many, many things you can adjust. So my general question for the CID design is that this is a setting will illustrate the importance of adaptation, okay? But then at the end, other than meeting the minimum kind of criteria, what do you need to look for to quote unquote optimize the trial design?
So first, and I think this was implied by your question, but I want to state it explicitly. All of the knob turning and adjustment has to occur before you finalize the design. And once you start the trial, it needs to be a pre-specified design. And when I call it a rigid design, but what's really rigid is the adaptation rules. The question of optimization begs the question of optimization for who? Whose utility function are we optimizing? And one of the things that, to me, makes this kind of design activity most rewarding is the fact that it's inherently multidisciplinary in nature. So when you're trying to decide how many interims, what the randomization ratio ought to be, or what your futility cutoff is, it really needs to be not a statistical question in isolation, but it needs to be a collaborative discussion among all of the stakeholders. And ideally, that involves investigators, clinicians, people, for example, in the blood banking community who would be using the results of this trial, maybe patient representatives and others. And it's really a consensus-based process when you have come up with a set of compromised settings for each of the knobs that balances the performance, statistical and otherwise, against the resource limitations, the complexity of the design, and complexity both from a statistical point of view and from an implementation point of view. So I think one of the things that I really enjoy about adaptive design is the fact that it naturally brings these collaborative groups together, and you actually get greater insights in what optimal looks like than you do if the statistician is working by themselves in designing the trial.
And also, just quickly follow up, seems like it's an overpowered design, right? And why do we do that?
So the reason we do that is because power is not the question, right? This is a duration finding trial. The value of this trial is not primarily dictated by whether or not there is a storage duration for which cold platelets are non-inferior. I think people who work in this area believe that there is. And in fact, the FDA issuing a guidance during the trial that says up to 14 days is okay would suggest the agency believes they are a reasonable option for the treatment of bleeding patients. And the guidance specifically separates, just to be clear, the use of platelets for bleeding versus the use of platelets for prophylaxis, that's a different clinical question. So the investigators and others clearly believe that the right answer for the question of the overall outcome of the trial is that we should be able to demonstrate non-inferiority because it's likely to exist. The question is, what is the maximum storage duration? And if the maximum storage duration is 21 days as opposed to 14, that has huge implications both in civilian and military settings for our ability to make this treatment available to patients who need them. That's where so the accuracy in duration finding was a primary design consideration.
Thanks. Steve, I saw your hand up. We have 1 minute, though. Is it very quick?
It's a quick comment. So, great study. I loved the graphics and tabulations of the simulations, made it crystal clear about controlling type 1 error, getting close to the true estimate. So, good job there. And then the other comment I make is this is a duration finding study, and I'm thinking about how does this maybe apply to dose finding studies and maybe more typical drug development, where maybe there's a safety issue with the drug and you just don't quite know how far to go. So let's do a low dose, collect some data, and then step it up. And at some point, we'll bump into the adverse event that people are worried about or whatnot. But I do think what you've presented is very good and perhaps could be generalized to, I'll say, typical drug development dose finding studies. So that's all.
Yeah, I don't think the mathematics knows whether X is time in days or milligrams. Thank you.
Thanks again, Dr. Lewis. So our next speaker is Dr. Karen Price from Eli Lilly, and she's going to be talking to us about a very interesting master protocol design for chronic pain indications.
All right, thank you so much. It is great to be here, and just to echo what Roger was saying, thank you for the invitation. Really excited and honored to represent this master protocol. I'm going to start first with some acknowledgments. These are several of my Lilly colleagues. Again, I'm honored to represent this on behalf of this team. It's been quite the journey. We submitted this, and so as I'm going to be talking today, I'll be emphasizing what it was like to go through the CID pilot program and the conversations that we had. But we were one of the first coming through, and so it's been several years' journey, and we've seen a lot of fruit out of this protocol, and so I'm excited to share that today. So what I'm going to do is start with an overview of the chronic pain master protocol, sort of set the stage and describe the design and the value of it. I will give some statistical details. In particular, as Dr. Scott had noted, we have an emphasis in the CID program around borrowing as well as simulations, so emphasizing some of the things that we considered during the discussions. I wanted to show an interactive tool that we utilized as part of the CID pilot conversations. I think that because simulations are so complex, as we've been talking about, and can slow reviews, to the extent that we can make them more interactive and help put into the hands of FDA reviewers the ability to explore some of these operating characteristics more efficiently, we'll see a lot of gains. And so I just wanted to show that and then talk a little bit about moving forward, some things that we need to consider to further advance CID. Okay, so in terms of CPMP, just wanted to start the importance of this case example. Chronic pain is a public health crisis and is one of the main reasons that people seek care. It's estimated that in the U.S., over 20% of adults live with some form of chronic pain. However, the probability of approval of novel analgesics that have completed phase 1 is significantly lower than for other novel drugs across other diseases. And furthermore, the current treatments are things such as opioids and nonsteroidal anti-inflammatory medications, which lack efficacy or have some safety concerns. So one of the things I think wanted to highlight here is that a lot of times when we think about CID, people do think about rare diseases, pediatrics, which was mentioned, and is certainly an important place for considering these types of designs. But obviously, in common disease states as well, we see a lot of value. And furthermore, is where we have a lot of data that we could really use and enhance these designs. So great setting for us to be as creative as possible to really help meet this unmet need for patients. There are a number of innovations that can be brought to bear when studying chronic pain. As you've heard what I'm going to focus on, and one of the things we decided to do was this master protocol. It served a number of, it helped us in a number of ways. One issue is oftentimes phase 2 studies will focus on one type of pain. Depending on the sponsor and how that progresses, if the pain type selected is maybe not efficacious, the drug may be abandoned, and so where it might have been successful in a different pain type, because it was only able to be tested in one, again, we see a lot of abandoned molecules. So what we wanted to be able to do is within the same protocol to test these 3 different pain types, and they were selected to be diabetic neuropathic pain, chronic lower back pain, and osteoarthritis pain. And so we were able to look at a variety of novel analgesics, and this resulted in reducing the size and cost of the studies versus had we done these independently, and or may not have even been able to test some of these drugs in these different pain types. So this slide summarizes the framework. You can see the 3 pain types coming down the rows there: osteoarthritis, chronic lower back pain, and diabetic peripheral neuropathic pain, really representing 3 different types of pain. So they were selected very purposefully, and a lot of discussion went into selecting those pain types. Across the columns, then you can see the different assets coming in. The protocol is sufficiently flexible that a given asset does not have to study all 3 pain types. Most have, I believe all have so far. It's not a requirement, but it is allowed, and we have that flexibility to look at the 3 pain types being enrolled simultaneously. Again, hard to depict here, but these are coming in. They can come in concurrently, or they may be coming in completely separately. There can be lag time and so forth. So it's an open protocol allowing those molecules to come through. I just wanted to share also kind of how we set up this protocol. So there were 3 tiers in this protocol. So we had the high-level master protocol that established the entry criteria for this master protocol, the randomization scheme, what the common the hypotheses that we were testing, the advanced statistical modeling and various operational features were included in this master protocol, it's really where we wanted to have the standardization that would be required. The disease state addenda, then that brought in the three different pain types. So anything that was unique, where maybe we had an additional measure to be evaluated for a certain pain type, that is represented in the second tier there. And then the intervention-specific appendices, so that's where the drug information came in. As I will talk about, we really wanted it to be as standard as possible, but if there was some unique feature for the drug, perhaps a drug-drug interaction or some tox consideration that at this moment in time needed to be different, that would be allowed. But we did have a governing body overseeing this protocol that would make those decisions about whether or not that modification for a specific intervention is necessary. And then this just shows the flow of how we sort of named things and how, again, this chronic pain master protocol, the three disease state addenda, and then we can add pain types. So it is flexible to allow for additional pain types to come in if we decide to do that. And then again, the intervention-specific elements. Okay, so next one I wanted to talk about then was what were some of the strategic considerations that we were thinking about, and highlighting the quote from Woodcock-Lavange paper from New England Journal. The common denominator here was we wanted to answer more questions more efficiently and in less time. So what we decided to do from a strategic perspective is we knew that we had several molecules that were going to be coming forward. We had a lot of opportunity in our portfolio, and so but that they were going to be coming in at different times, and we wanted to be able to make the best decisions about which ones to move forward and in what pain types. This is a phase 2 proof of concept study only, and so we're very specific that it is about hitting it hard, understanding is there a signal or not, and proving that concept. Following this would then be more robust dose finding, ultimately moving on to phase 3. So this particular design was not constrained by registration requirements, although of course we wanted to maximize transferability to phase 3. And as I mentioned, we would have phase 2B to follow. We did limit sites to North America, and then, as I mentioned, that the master protocol structure is established and allowing for that flexibility within the ISAs as needed. Some key features of this master protocol: all of the ISAs have the same scale. It's the numerical rating scale. This is the primary endpoint across all three pain types and across all molecules, which is really important. Again, pain is a very subjective indication, and so we had the same scale, we had the same sites, we had the same. There was not a washout period, everything was consistent, which really helped to remove some of the confounding factors that often enter when we're looking at independent pain trials. There were other scales: physical functioning, emotional functioning, and so forth. We had standard data collection, similar visit schedules, and then, as I had mentioned, there was a master protocol team established to analyze the efficacy data to make decisions about, as I mentioned, any difference that an intervention might need. Is that really necessary to make the difference? Because we wanted to keep things as standard as we could while allowing proper flexibility where it was absolutely necessary. Primary efficacy analysis is a Bayesian mixed model repeated measures. This was the primary efficacy analysis. Again, it was using the NRS, and what I wanted to emphasize here is the primary critical success factor is in the framework, as you can see in that sort of second sub-bullet, the probability that the treatment difference is less than an effective interest, and it's less than because negative is good here, so more negative. The probability that difference exceeds a threshold that's, again, established before the trial starts. And so we had this framework in place at the master protocol level, but we recognize that as an intervention comes in, The effect of interest might change over time. In particular, if there are molecules that are successful, then we need something more, even higher perhaps. But then also that probability threshold may evolve over time. So we allowed that flexibility and just insist that it's specified prior to that specific asset enters into or starts in their ISAs. All right. And some of this I've talked about, but just maybe to restate and touch on a couple of additional things. You know, anytime working on a master protocol, it really is this tension between what is going to be standard and what is allowed to be flexible. And it was very important, given, as I mentioned, the subjective nature of pain, that we have as much standard as is possible. So again, same primary endpoint. We had the same probability of getting placebo, so 33% randomized to placebo across the ISAs. There is a double-blind period of 8 weeks, but on the other hand, if we have a molecule that's coming in that is not able to be studied for 8 weeks, we had the flexibility to allow it to be studied for 4 weeks, and then it would be double-blind placebo for the final 4 weeks, just maintaining that same treatment duration. Again, common visit schedule and inclusion criteria, but from a flexibility, the ISA can specify sample size. The amount and type of borrowing will obviously evolve over the course of the study, because as we're gaining more information, we'll now have new information that we can utilize. And then, as I mentioned, there can be inclusion-exclusion changes or scales, visits added, but governed by one group to make sure that it is necessary. There's been a number of statistical benefits from this. I think probably this top one is one of the most important ones, where we're able to directly compare within and between pain types. We had an advisory board before the master protocol formally started, and one of the members was commenting how often do we wish a drug was in the same protocol and we didn't have to rely on meta-analysis. And that's exactly what we were looking to achieve, that we can do indirect meta-analysis or other forms of meta-analysis, but especially in the context of pain, which is subjective, we wanted to reduce a lot of the confounding factors. And it was a lot of enthusiasm from our FDA counterparts during the CID meetings about our ability to do this across pain types. Again, a lot of times these trials will utilize different endpoints, so maybe it's a VAS. How does that compare to the NRS? It's therefore hard from a meta-analysis standpoint to do that, and or it's lacking consistency in collection of safety and biomarker. And ultimately, the master protocol affords the opportunity to reduce sample size in both the active and placebo arms. And so thus far, we have seen, as I mentioned, a great deal of impact from this trial, including reduction in cost, reducing time from, say, protocol approval to when a first patient is dosed, time to data lock, time to decisions, and enrollment time, completed 12 proof-of-concept studies in 38 months, validated 3 novel targets, and this is much shorter than what we would see independently, but is also hard to fully compare because, again, often we may not get all 3 pain types because if the first one isn't successful, it may be abandoned. Okay, so let me take a little bit of time then and go into some of the statistical details. As I mentioned, I'll focus on the borrowing conversations we had and the simulation. For this master protocol, there were 3 main sources of borrowing that we considered. The first source was not unique to the master protocol, can always be considered. So we spent more time talking about borrowing from these second 2, the ability to borrow from placebo information from another ISA within a pain type, and then the opportunity to borrow treatment effect information between pain types. I'm not going to go into a lot of detail on the borrowing approaches. You'll hear more, I believe, from the next speaker on how some of these approaches work. What I will mention is that we typically are thinking about 2 main buckets of borrowing approaches. one being static, and that includes things like pooling or power priors. Dynamic borrowing, on the other hand, things like hierarchical modeling, mixture priors, or commensurate priors. And there's an appeal for dynamic borrowing where what happens is that if the incoming data is consistent with historical data, it will borrow more. If it looks different, it will borrow less. And toward the end of my presentation, I'll show an example where sort of pooling versus the hierarchical modeling, you can see that when the new data comes in and is different. So what we ended up doing, and as you'll see later with the tool, is we spent a lot of time talking through with FDA and comparing things like pooling or power priors, more static, on the other extreme separate, so not borrowing at all, and then something in quote unquote in between, and so say the hierarchical modeling approach. So that was where we spent a lot of time understanding how would decisions differ based on incoming data. When the rubber hit the road, or when actually the reality of conducting this trial and working with teams and data coming in, there were a number of challenges that were encountered. So certainly things we thought about prior to the design, but then again, this is an ongoing trial, and assets are coming in and going out, and things change. And so some of this we've sort of figured out. Other of it we haven't. So I think some of these are still outstanding questions. As I mentioned, when inclusion-exclusion criteria change necessarily, how does that impact the ability to borrow? Do we use pooled placebo or ISA in a safety review? We did allow for patients to repeat enroll into later ISAs after a proper washout period. What do you do with that patient's information from earlier? Is it borrowable? And what does that even mean? So there was also hesitancy to borrow from some team members, so there's a mixed feeling on whether or not to borrow. And I think similarly at FDA, right, when we encountered there are different viewpoints on the what should be borrowed and whether or not to borrow. Again, what is the best approach to borrow? Spent a lot of time talking about should we borrow across pain types, and then also thinking about placebo expectation bias and how does that change over time and affect our ability to borrow. So those were the borrowing approaches. Again, we spent a lot of time simulating these trials, or this master protocol, and then looking at different ways the ISAs would come in and the type of data that would be coming in. Some of the key factors so we did a lot of simulation as part of CID before the trial started, and then as an asset comes in, additional simulations are conducted, given that now we have new data that's been completed from earlier ISAs. But before even the master protocol started, we looked at different scenarios, thinking about how much placebo data could be available from either completed or ongoing ISAs. What would the different treatment effects between pain types that we could observe, and how does that affect how it's being borrowed? We looked at scenarios where there could be placebo drift that could occur. What would we do and how would we handle that? And then we knew that there was a possibility of having a different route of administration. And so if something were, say, injected or oral or given more frequently, those are things that can affect the subjective endpoint of pain. So how would we handle that? So we spent time looking at different scenarios and working through those with the FDA as part of our conversations. And we did think about fixed versus longitudinal time point settings. This slide just summarizes the types of things that we were looking at in the CID conversations, very similar, I think, to quite a bit you heard in the earlier talk: power, false positive, bias, standard error, understanding the operating characteristics, as I mentioned, when the underlying true placebo response shifts over time. And again, that could be due to some new drug that is approved. So we what could happen to affect that true placebo response. Benefits of power increase or sample size reduction. we explored the various ISA initiation and lag times and enrollment dropout rates, as well as the quantity of data that would be available. All right, and then the final thing I wanted to touch on on the simulation front before concluding here is I wanted to touch on this R-Shiny application that was a feature of our CID pilot program conversations. And as I mentioned at the beginning, the simulations there's a lot of simulation output, and typically it's provided in paper. We may not provide a scenario the FDA is interested in, and then there's an iterative nature there where we're providing that. And so what we really wanted to do was to build something that could be more interactive, both for us, but also for FDA, to evaluate this design, reduce the amount of paper that's required, and provide more interactive visualizations. And with the ultimate goal that I think is, and maybe part of what we'll talk about more this afternoon, is the importance of modernizing this collaboration to help speed the review of simulations and just to continue to use the best computation we have, the best tools and technology to make this easier on everyone. So we can really get to the heart of the matter and really have those cross-functional conversations that Dr. Lewis was talking about. That's really where we want to be spending our time, not trying to make sure we're reading a table correctly, but rather really getting into those great cross-functional discussions. And so more interactive simulation output, we think, could be very helpful. This application, you'll see in a moment, and I'm just have a short video demo. It's more just to show what we mean. The specific application has two main parts. One is if you have, you may want to look at a single realization of a master protocol. So suppose you're really interested in what would be the output, the inference made in terms of the probability of achieving a CSF under a certain given single scenario. So it fits a model. You can look at different amounts of historical data that would be available. And so that's one part. And then the other part is it simulates multiple trials, again, to evaluate those operating characteristics. And so the user can enter data from completed ISAs, simulate future ISAs, vary the model prior, and so forth. This is just an example. Again, actually what it would look like can vary. Again, the intent here is not to go through this whole thing. I'm about to start it in just a minute, and you'll see the interactive nature of it. But the idea here is, again, to give the user the ability to look at scenarios or look at situations they may be interested in. Again, this first part is the model and the priors, and so you can see there's click down boxes to specify the model, prior distribution for tau for the hierarchical model. That's on the placebo effect, and there's a treatment effect prior as well. And then, as I mentioned, the user can enter the example data, and it will run and provide the posterior distributions, and you can zoom in. Here you're seeing zooming into and looking at the different borrowing approaches. Further down, we can look at the probability of achieving the critical success factor. Maybe the user wants to look at different effects of interest or probability thresholds. In the green there, I think what's really nice about this is you can see which borrowing approaches meet the CSF versus those that don't. So you can see where a different borrowing approach is yielding a different outcome. There are some diagnostics that are included here. So we can pick the parameter you're interested, check the diagnostics, and then you do get the actual parameter estimates table with posterior means, standard deviations, and so forth. Now we're going to look at simulations. So here we have simulation scenarios that have been included, and this is quite, the user can play around with this and then run the simulations. Kermit the Frog is going to help us run those in the background and then provide the output to the user. Again, these scenarios on the upper left-hand side, those are what you can enter different scenarios that you may be interested in. Here, we're just selecting a parameter, and again, you can determine which parameters you're most interested in, and then look at the operating characteristics. And then finally, the bottom portion here, and this is where I'll touch on something I mentioned earlier, we're looking at, and you have different borrowing approaches, and we're looking at the probability of achieving the CSF. In this case, we did use a scenario where the historical placebo information was very different from the incoming placebo information. And so as you click through the different borrowing approaches, you'll see the differences. And then as we scroll down here, we're looking across all of the borrowing approaches that were considered. You can see in that middle part that pooling is resulting in a very different outcome as versus the hierarchical, which is on the left, or separate, which is on the right, showing us that the hierarchical model is in fact borrowing less in this case because that new data is different. So just again, an interesting scenario and the types of things that can be evaluated through an app like this. And then a couple things finally, to conclude here. Generally, our experience with the CID pilot program, there were very positive interactions between Lilly and the FDA, absolutely improved the master protocol, and we think advanced how we're thinking about borrowing of information, master protocols, and simulations. So we, again, very collaborative, progressed how we could communicate these methods, simulation plans, and results. And because they were so positive, we do need to continue to have these pathways and avenues to have these types of collaborative discussions, really to the extent that we can have some informal pathways as well, which this was, while it was formal, it had a more informal feeling because we were talking about the different methods, talking about what data sources might be useful, these sorts of things. Again, I touched on the R-Shiny collaboration. In terms of opportunities for improvement, I think our biggest recommendations would be the timeline is long. So it was actually, because of the situation going on at Lilly with the assets that were coming in, we did not delay anything by doing this, but if in normal cases, we would not have been able to probably go this route because it is a timely process. So we knew well in advance what was coming and therefore sort of had the luxury, if you will, of going through this. We do think that it would be great to have also opportunities, especially for something like a master protocol, but any design as data comes in, what are we learning? And I realize this forum is part of that, but having some more conversations with sponsor and FDA continuing, we think would be very helpful to continue to share learnings, and then ensuring that we continue to have consistency in those attendees. And so finally, what are some things that we can think about moving forward, and maybe part of what we'll talk about, how do we continue to share learnings across divisions? We have taken additional master protocols into other divisions, and there are different types of feedback that's being provided, and so how can we have more consistent across divisions in terms of the feedback and the expectations? I mentioned the improving the infrastructure, continuing to look for cutting-edge technology to enable us to be faster as we evaluate simulation results and interactivity. I've already touched on the meeting schedules that can accommodate the speed that's needed. Improving the education of statisticians in medical, both, again, in sponsors as well as at FDA. And then some other things that will continue to play into how do we advance these innovative designs, what's the role of AI/ML, other new technologies, and then how does the fact that there's so much move toward decentralized trials and digital health technologies, how does that affect how we borrow, if we borrow these types of designs? So things to think about. So with that, I will close. Thank you.
Thanks so much, Dr. Price. Would anyone on the panel like to comment or have a question? Dr. Rupert?
Well, that's complex and innovative. So I'm just wondering, you had different probability thresholds and effect sizes in your decision criteria. I'm just wondering if you can talk a little bit more about, so how are decisions made? You know, one asset completes osteoarthritis before it completes lower back pain, and or they all complete at the same time in one disease state. You meet the critical success factor. One, you fail miserably. One's right on the borderline. Can you talk about the complexities of the decision making about what goes forward or what gets held back, or of these three assets, this one has the biggest probability of having a clinically meaningful effect? I was just wondering, it's not only complex in its setup and the analysis options, but also in the decision-making framework. So a little bit about how that worked out, or how it is working out, I should say.
Yeah, sure. So yes, it is complicated. I think this is where having that centralized group has been vitally important. So we have one group who's overseen this master protocol, and of course there is some rotation, but you have some members who've consistently seen this protocol, know it inside and out. So that has helped. Typically, in terms of your question on the timing, there is some lag that can happen where, like you said, maybe OA pain finishes and then a little bit later chronic lower back pain. But we know well in advance when these things will happen, and so there are decisions made before we start seeing data. Are we going to pull the trigger on, if it's highly positive, are we going to go pull the trigger on the next phase 2B, or are we going to wait for the data? So that is the importance of making these decisions before we see data. I cannot say that enough. You know, once you start seeing data, then it gets very confusing. So we have basically a pre-planned, here's how it's going to be communicated, here's how we're going to make the decision, what's the level of efficacy required in order to do that phase 2B versus, so there could even be different thresholds for different decisions. So I think, like I said, the main thing would be pre-planning that, understanding the timing. If it's a few weeks, it's probably worth waiting. If it's going to be a while, then maybe we need to do something different. So really, like I said, the centralized group, pre-planning, and understanding how does it impact. The last thing I'll say is sometimes it may be we say, okay, we think we're going to move this one forward, so we start sort of planning that trial, but we're not going to do too much until the next thing comes out. A lot of things to think about.
Dr. Lee?
Yes, hi, Karen. I really like your shiny egg.
Thank you.
Okay, and I think it really is a very good tool to enhance the collaboration and even communication, with FDA. So the question I have is that I'm sure this is not public available yet, right? But at a certain point, it would be great that if you can turn it to a more general purpose, kind of a tool as an education tool, and maybe publish it, and so that people can learn how to do this. So any plan for that?
Well, so I think there are, first of all, I should acknowledge people like Eric Nance and Michael Songson for working on that. Eric Nance is very active in the R community and is working with counterparts at FDA to really understand what technology is required. And so it's not just Lilly, right? It would be, these are more scientific working group type opportunities where they're looking to see what technology does the FDA have, what does technology would the FDA need, because that could certainly be part of some future PDUFA negotiation or whatever the case may be, as we need to advance this to allow for both FDA and sponsors to have it. So yes, a lot going on, and there are things that could be available to do this, but I appreciate the feedback, and we should continue to try to push that.
Dr. Lewis.
So on the same theme, I think one of the more challenging discussions people often have in discussing designs that use borrowing has to do with the choice of the hyperprior that's used for dynamic borrowing within a hierarchical model. And I really like that you were able to show with the app the performance in the specific situation where the historical data did not well match the data that was coming in currently. But there's the challenge in picking the hyperprior is that you have to account both for that situation where you have discordant information and the opposite situation which might occur where the information is highly concordant. I'm wondering whether you've thought of a display that shows both of those simultaneously so a person can just glance sort of at a prior picking dashboard and sort of see where it does well and where it doesn't. Because we've struggled, frankly, in figuring out how to communicate the implication of these choices to these collaborative design teams and make sure that we have a prior that reflects the best balance between performance when you have concordant data and performance when you have discordant data.
So great. We'll think about it, I guess. Currently, the scenario is entered. We've evaluated another scenario. And so we'd look at, to your point, we'd look at everything looks great, things don't look so great. But to do that all in one, we'll think about it and get something in there.
Yeah, I guess I'm also a fan of the Shiny app. What I particularly liked was when you could have no borrowing and get the posterior distribution for that compared to various levels of borrowing. So it sort of made visually and precisely how much, the borrowing was contributing. I had a comment, I guess, inspired by an earlier question. So you have a particular drug and you might be evaluating in back and osteoarthritis, and you might you might stop randomizing in the back but continue osteoarthritis. Is that how it might play out? Or do you just keep randomizing until you get the answer for that drug?
So what happens, what I think you're asking, so there's a pre-planned sample size for each pain type. And so what you were seeing there, those were all simulations or made up examples. And then the trial, let's say that we're going to have a couple of hundred patients in a given ISA, randomized 2-to-1. We enroll until that is done for that ISA. But now, if osteoarthritis is ahead of chronic lower back pain, we don't keep enrolling osteoarthritis. Once it's done, it would stop. So that's why they can finish differently. Well,
then just a minor comment on, so osteoarthritis might stop and you continue enrolling. You could, in theory, sort of borrow into the future where you get placebo group for osteoarthritis that wasn't concurrently enrolled. So
that is correct.
Yeah.
Yes. Yep, that's exactly right.
So we are almost at time. Dr. Price, just one quick question from me. I hope it's quick. You mentioned some internal hesitancy to borrowing. What was the nature of that hesitancy? Was it scientific or strategic or regulatory?
I believe it's probably a combination. In this case, because this is a proof of concept, I don't think it was so much regulatory concern. This would be more if the borrowing changes the decision, I'm concerned about that. So it's more like a personal, maybe not fully trusting the borrowing.
Thanks. Yeah. It's not that dissimilar to some people internally.
Yes. Yeah.
Okay. All right. Thanks, everybody, for 2 great talks and for your attention and for the questions. We now have a 15-minute break, and we will resume with Dr. Peng's talk at 10:50. Thanks. So thanks, everybody, for coming back after the break. We'll give people a moment to get settled. And in the meantime, I'll introduce our 3rd and final case study for the morning, and then it will be lunch. Herb Peng from Genentech Roche is going to be talking about their CID case study involving the use of a hybrid control in diffuse B-cell lymphoma. Dr. Pang.
Thank you, John, and thanks everyone for coming. And also, just like Karen and Roger, I would like to thank the organizers for the invitation. So today, as mentioned by John, we'll talk about a case study of hybrid control design in diffuse B-cell lymphoma. First, I'll briefly go over some introduction about the study designs with external controls, and follow up with very shortly talking about the CID pilot program as well as the timeline. We'll go into details about the Genentech Roche CID pilot. After that, we'll also cover hybrid control ongoing research. And I'll also conclude with a summary. As you may know, for study designs using external controls, there could be different ways you can go about doing it. On the left-hand side, there's a threshold crossing benchmarking approach, where in the external control arm, you would have applied the randomized control trial inclusion and exclusion criteria to have a restricted external control arm, and then you would read out the outcome. In the middle one, you have a single arm external control study, where you have the external experimental arm in combination with the external control arm, and then forming the cohort, but then you would do covariate balancing or adjustments so that you can make the comparison between the two. And finally, on the right-hand side, you have a hybrid external control design, which is the one that we would talk about today. You have 200, for example, randomized control trial subjects, and then you split it into, for example, 3-to-1 randomization, and then having 150 in the experimental arm and then 50 in the control arm. But unlike the typical randomized control trial setting, you also incorporate and augment the control arm with external controls. In this case, for example, you have roughly 100. And then you would run this trial and read the outcome. Thank you. In considering the choice of when to do a hybrid design, you may think of two extremes. On the left-hand side, you have a randomized clinical control trial, and on the right-hand side, you have a fully external control trial. One factor is whether there's a medical need. And for the randomized control trial setting, you can think of it as if you have an effective control available, it could be a good choice. On the other hand, for the fully external control trial, it could be that there's clear med need, however, there's no effective control available, and then it's kind of like a spectrum, and a hybrid design would fall in between. For the target indication on the left-hand side, when you think about a randomized control trial setting, you may think of things that are more tied to first label or new broad line extension. And then for the fully external control, you may have line extension in similar indication or indication with well-documented standard of care. Choice of endpoint is also an important consideration. So for the randomized control trial setting, you may consider whether there's a specific endpoint that you don't have data readily available from external sources, then you would probably, it's wise to choose the randomized control design. On the other hand, if you want to do the fully external control part, you want to have a robust endpoint where you have data available from the external sources. For example, overall survival, etc. So again, the hybrid design would fall in between the two. In terms of anticipated effect size is also an important consideration. For the randomized control setting, you may think about having it as in cases where you have modest effect size anticipated based on some observed prior information. On the other hand, for the fully external control study, you may want to have more compelling effect size. And then finally, for population size, Having a large population and no challenges in recruitment is probably an important factor. You want to get the trial completed on time and within a reasonable time frame, so you can consider an LCT. On the other hand, for a fully external control study, you may have potential issues with recruitment or some ethical challenges, then you would choose a fully external control. And again, for the hybrid design, it's somewhere falling in between. So as we may know, there are different potential biases that can come from the use of external control sources. These include, but not limited to this set, such as selection bias, where patients enrolled in clinical trials are different in some ways compared to patients that are treated in clinical practice. Other biases could include calendar time bias, where patients treated in the past do differently than those treated today. Regional bias, if you have enrollment of subjects not just within the U.S., but in other parts of the world, that patients coming from different regions could have variations. Assessment bias, knowledge of therapy that can influence the outcome assessment. And study bias would be patients in clinical trials, they have different outcomes than in clinical practice. For example, placebo effect or different care. As I mentioned, this is not an exhaustive list, so there are other biases that you also need to consider. I think the FDA also had a guidance that provides some knowledge about these potential biases, but framed under more the design setting, which is also very important in terms of how to think about these biases. Some important thinking going behind how to mitigate potential biases to understand the Pocock criteria, which was developed many years ago. Receiving a precisely defined standard treatment, the same as randomized controls, is one important factor. Being part of a recent clinical study which contained the same requirements for patient eligibility is another one. Methods of treatment evaluation should be the same. Previous study must have been performed in the same organization with largely the same investigators, as well as there must be no other indications leading one to expect the different results between the randomized and the controls that are historical. The distributions of important patient characteristics should be comparable in those in the new study. I think earlier Karen also touched upon about this, about the dynamic borrowing design. So let's take a look at this illustration on the left-hand side. At the top, or the extreme top, is the no borrowing scenario where you only use the randomized clinical trial. At the other extreme is the bottom, which is borrow more, and that would be the case where you consider full borrowing, where you just simply pool the two controls together and estimating the treatment effect. And dynamic borrowing is somewhere that falls between these two extremes, so it belongs to a spectrum. And even within dynamic borrowing, you can consider different types of prior that would be more, for example, skeptical prior would be more conservative. And then you can have a more aggressive prior if you have more being more optimistic on the external control. And later on, we'll illustrate in our case how you consider choosing between these two. The Bayesian method presents a natural way to handle combination of data. External trial data can be used to setting up the study prior, and dynamic borrowing framework, as mentioned, could be an important allowing you to kind of understand the difference between the internal and external control. So when you have more differences between internal and external controls, you would likely have the prior which is more flat. So in those instances, you would borrow less. And then on the other hand, if you have more trust, then you have a prior that has more like weight, and then that would provide a borrowing scenario of borrowing more. So that's the commensurate prior. In the publication that was published about 10 years ago, they discussed the dynamic borrowing methods were able to achieve similar power gains, which is the color in green and blue. But then in the full borrowing scenario, which is more extreme that we described earlier, which is in red, it has more type 1 error inflation. So the dynamic borrowing method can achieve similar power while having better type 1 error control than full borrowing. So I won't go into detail about the CID program here because John already described it very well. I just want to mention that it has been a great opportunity and also a wonderful experience for us to join the program. This is the typical timeline of the CID program and the process and how things happen. I think unlike the scenario that Karen had, I think in our case, we actually needed more time in the end to do the simulation. So it was actually us asking the FDA to allow us to provide a more comprehensive understanding of the method for the second meeting. So in the FDA, were very collaborative and flexible in allowing us to meet later. Why innovative design was needed for our case? Unmet medical need, as described earlier, in certain subgroup of DLBCL was the case. DLBCL is most common non-Hodgkin's lymphoma worldwide, with 25,000 newly diagnosed patients in the United States annually. And standard of care for first-line DLBCL has been established many years ago and is well characterized and well understood. Patients in certain subgroup of DLBCL have a poor prognosis and consequently a high unmet medical need. So borrowing patients from control arm of another study can help us enroll fewer patients in the control regimen, allow us to shorten the study time, and also conduct more efficient trial by sharing control between trials. Here was the timeline of the brief phase 3 development for the first-line DLBCL and pathway to the CID pilot. And initially, we received encouraging data about the phase 2 study compared to historical R-CHOP control, especially in the biomarker-positive patient group. Other control can potentially limit the number of new patients exposed to well-established standard of care. FDA Type C meeting on proposed phase 3 in biomarker positive of experimental plus R-CHOP versus R-CHOP, 3-to-1 randomization plus externally borrowed control was selected from internal study. And the agency recommended actually the primary analysis population and analysis plan to be on a randomized trial without an external control. The analysis population can be used for supportive analyses. The focus of the updated design with the external control was for the secondary endpoint overall survival, because overall survival is a clinically meaningful endpoint with minimal ambiguity in its assessment. So then we joined the FDA CID program, which really provided a great opportunity for us to build on utilizing these external controls and discussion, and also within a very collaborative framework. So eventually, we came up with this design where we would randomize subjects 2-to-1 with the novel combo with 280 subjects and the R-CHOP group of about 140. And then we supplemented the external controls with about internal controls with the external controls of about 100 subjects. As I mentioned before, the primary endpoint for the study is PFS, but that was assessed only with the randomized subjects. And the key secondary endpoint is where we actually combined the randomized subjects and also augmented the internal control arm with the matched external controls. The external control patients were selected from a contemporary ongoing internal clinical trial, and the intent to support early OS analysis at the time of primary PFS analysis. So that's the shortening the time to get the readout. Randomized study with external control arm used matched external controls through, as we mentioned before, dynamic borrowing. So the rationale for sources of external control arm. There's a prospective plan to select external controls from an ongoing contemporary internal randomized controlled clinical trials, consistent with the eligibility criteria planned, aims to target similar and investigators' sites. Overall survival is, as mentioned before, also very clear and clinically meaningful. So 5 out of 6 proposed criteria with the POCA criteria that was mentioned earlier were met. So what does our kind of analysis of flow diagram look like? So the first one is to look at control comparability evaluation by applying inclusion-exclusion criteria, as well as flagging baseline factors that have significant differences between internal and external trials. The next step was to utilize propensity score matching to match patients between internal and external control trials using propensity score matching, which enhances the covariate balance between the two groups by filtering out unmatched patients. Finally, we applied the Bayesian dynamic borrowing method, which automatically downweights external control based on the agreement between internal and external controls. And again, as we mentioned, we provide inference for the overall survival for the borrowing scenario, and sensitivity analysis would follow the main analysis. I won't go into all the details about the simulations, but briefly we'll cover kind of at the high level what we looked at. And the main goal of the simulation scenario is to evaluate the proposed statistical method, which is a combination of propensity score matching with Bayesian comparator prior approach. We examined a few things to understand the operating characteristics, including varying magnitude of differences in baseline characteristics, which we will see in the coming slide, as well as the different choices of prior, which may influence the degree of borrowing. We'll, at high level, cover how we looked at the violation of various assumptions. So on the left-hand side, you see a plot of type 1 error. And at the bottom, you have different types of scenarios. So there's a scenario on the bottom left, which is the no difference, followed by the moderate difference between the internal and the external controls. And also the scenario where you have a large difference, plus the benchmark, which is the no borrowing case. So as you can see, the no full borrowing case would have the highest type 1 error inflation, while the dynamic borrowing with half Cauchy is doing better in the type 1 error control. And in terms of the no borrowing reference, is not too far from that. As for the power gain, they're also shown on the right-hand side with the different scenarios, no difference, moderate difference, and large difference. And of course, given that the borrowing case all demonstrated an increase in power gain, but we also need to take into account the type 1 error inflation. And as we can see, the dynamic borrowing with half Cauchy does have a decent power gain. In terms of violation of various assumptions, we won't cover the results in detail here, but what we looked at include understanding and simulating the scenarios where we have, for example, observed a measured confounder, as well as understanding if the survival curve distribution is different from the assumptions that was made. And we also looked at if there's a nonlinear or non-additive defect model, how does the operating characteristics performed. So in conclusion, we found that in general, the dynamic Bayesian with the conservative half Cauchy prior was able to have a good average error rate, weighted type 1 error rate, and a slightly inflated maximum type 1 error rate, but is the most conservative one that we observed, much better than the aggressive as well as the full Bayesian scenario. So what were some feedback on this, the potential of using OS with external controls? There are several aspects we learned, including model assumptions assessment, which is standard analysis typically requires fewer assumptions. So these Bayesian scenarios have more assumptions, can be less standard, so you need to understand and assess them, just like as we demonstrated with the violation of assumptions simulations. The need for pre-specification, I think earlier speakers also alluded to that. And then another consideration unique to this case would be what could hamper inclusion of overall survival in the label. For example, whether the model assumptions appear to be met, any outlying subgroup effects, is the endpoint credibly captured or not, overall conduct of study, missing data, as well as, for example, whether the baseline characteristics are the same. In addition to statistical considerations, there are other considerations that you need to think about, which include is the analysis of summary of analysis clear? Can it be interpreted by clinicians, as well as would it provide valuable information? With these more novel designs, it's unlike the typical case where you can decide on parameters and have fixed scenarios. So you need to do more extensive simulations. And for the case that we did with the CID program, we actually had many scenarios per FDA meeting. So we have a couple of meetings, and each of them has more than 20 scenarios that we investigated. So the implications would be that we have to plan early, allocate sufficient time and resource, as well as having, for example, software being available. So right now we have this open source software. I think Jack asked a question earlier about having open source resources so that other people in the industry can also benefit from it. So PS Pro 2 is one such example. And of course, internally, we need help from other Roche statistical software engineering team and methods experts. So learnings from the CID program certainly helped us a lot. And a related initiative that we have that we'll cover very briefly in the coming few slides is another FDA initiative, which is the U01 grant. So in 2020, FDA awarded 4 grants through the U01 mechanism for the exploring use of real-world data to generate real-world evidence in regulatory decision-making. And I think they actually have a new batch that came out last year as well. And if you're interested in learning more and you're a member of the ASA BioOp section, there's actually a session this Friday that would actually have some speakers that have been awarded these grants. So this is an ongoing research, and which is entitled Applying Novel Statistical Approaches to Develop Decision Framework for Hybrid Randomized Clinical Trial Design, and combining internal controls with patients from real-world data sources. So this is in collaboration with University of North Carolina. One of our research work has been recently published utilizing something different than what we presented in the CID program, which is a case-weight-specific power prior method. So as we saw in the dynamic Born case that we actually have this maybe assessing the agreement between internal and external controls and then putting a weight, but the weight is the same across all subjects. So this particular approach would actually have a case-specific weight. And it's soon to be available online, but there's also an archived version of this paper. I think this point was also touched upon by Jack when he asked a question about, or talking about the turning the different knobs, different ways, different parameters that can go into it. So one of the questions that came up during our discussion of the grant that we have monthly meeting with the FDA on is the randomization ratio. So how do you choose and understand that? So in this paper, I won't go into all the detail about it, but essentially for the scenario very similar to the CID case that we have, we try to understand what's the impact of the various parameters on, for example, the internal and external control ratio. So we discovered that the internal-external control ratios is one important aspect, but also the randomization ratio can also affect how the operating characteristics can behave in terms of type 1 error and power. So this work is published in the ASA Bio report. This work is another piece that actually something that came up from our CID collaboration. And when we were doing the step about the before the dynamic borrowing, FDA suggested that we should use propensity score matching. One of the reasons for that is that there's not much literature on how does it work with other ways of handling, for example, propensity score weighting or even covariate adjustment. So in this work, we actually investigated also the operating characteristics, how different approaches in handling covariates can have an impact on the design. So there's ongoing research on this topic, and what we covered mostly today are tied to more time to event outcome, but there are other outcomes that I think in Karen's talk and also in Roger's talk that it was covered that there are different outcomes that, for example, rare diseases, pediatric outcomes could have different endpoints and different characteristics. So oncology was the application today, but we also have, for example, other settings as well. So in summary, the CID pilot program, we facilitated a very collaborative scientific discussion with the FDA. And we had alignment on critical concepts, design proposals to boost confidence in future designs and outcomes. Agency also demonstrated openness to the proposed design with external controls while providing key feedback. As we learned, early sponsor and health authority engagement is paramount when we want to do these novel trial designs. And successful adoption of novel innovative designs also requires collaborative effort between health authorities, academics, and industry, as you see from the panel that we have today. So we highlighted one example of how the methods work has been used to fill the research gap. So this work is takes a team effort, and for the preparation of the CID event and also the CID program as well as the grant, these are the colleagues that have helped us. And thank you for your kind attention. Yeah.
Thanks so much, Dr. Pang. I'll turn to the panel to see if anybody has any questions or comments. Dr. Hubbard.
Thanks for that really nice example of a hybrid controlled trial and all the sort of many considerations that go into how you put it together. I had a question about a specific element of it, which was the choice of the number of external controls. sometimes we're in settings where you have a very large pool available from a registry or other external source. I'm not sure that was the case in your example, but in general, how would you go about thinking about the considerations there, the practical constraints, the effects on the operating characteristics, and then also I think there's sort of a gut feeling that you just sort of don't want to have too many external controls, and how do you weigh those things?
Thank you, Rebecca, for this important question. I think this is one thing that probably in our talk we didn't cover as much in detail. One of the reasons is that in the CID program and what we have for the case, we actually have a concurrent ongoing trial, which makes it much easier in terms of having high-quality data that really follows the POCOC criteria. I think in the case of if you have a scenario where you can borrow a lot more, but you may be more skeptical about the quality. I think data is so important, right? The quality of data. So having the screening of inclusion and exclusion criteria to make sure the two groups are aligned, the internal control and what you use to augment it, is extremely important. So a filtering step is certainly important, but how you go about doing the filtering, there are many different approaches to do that. So one such would be maybe propensity score-based approaches, but there could be other ones, like setting up ways to make sure that you just exclude certain subjects, right? Because there are some differences in terms of calendar time biases, like you want to ensure that things are aligned in terms of when you enroll the subjects. So just like you have to consider all the biases that may be involved and read through, for example, the paper we discussed, but also the FDA guidance on how they evaluate, right, the subjects in the end. Because in the end, if there are more uncertainty, there's also a risk, right, to reading out something that things are less aligned. But I think we are very fortunate in our case that we can borrow from something that's concurrent and ongoing, and which is sort of also blinded, right? So I think that scenario is very ideal. So I think that's also why we were selected, because this is one of the better scenarios that you can have. Yeah. So thank you for the question.
Dr. Pullman.
Yeah, just to pick up on that comment. So I guess you were underpowered for overall survival, and so, like in theory, you could at the end of the study look at how many deaths you have in the randomized trial, and then do a power analysis to decide on how big the external control could be. It could be that you have enough deaths that you don't even need it. That's probably unlikely. But anyway, you could calibrate it to answer, to formulate it with a power, based on a power analysis.
Yeah, thank you, Dean, for a great comment. So yeah, I think in some scenarios, definitely that could be a good choice. So for example, you can just understand whether it's already enough. I think in our case with the DLBCL population, the overall survival actually takes a long time to mature. So even with the borrowing, I think we are not really at 80%. So it's more like 60 to 70%. So that's one of the reasons why in this particular case, the borrowing could be a very good approach. And plus it's a secondary endpoint, less sensitive to if it were to be a primary one. So, but I think the idea of maybe having something pre-specified that you can adapt whether you borrow or not could be a good approach. But in addition to the simulations that needs to be run, but also the other scenario, how to go about thinking about that, right? So, but that's a good idea, yeah. Thank you.
Dr. Lewis, then Dr. Ruberg.
So I think one of the challenges in dynamic borrowing of control data has to do with how you quantify the benefit of the approach and communicate that benefit to decision makers and others involved in deciding what the approach is you're going to take. So I'm curious in this setting, how did you quantify what you gained through this approach, since obviously a lot of work had to go into it, and how effective was that communication?
Yeah, so that's a great question, and of course, communicating the results was like even the simulation settings and the results is also very important. So in terms of understanding whether I think one of the key things we look at is really the type 1 error control. I think FDA, as you know, is also very keen on making sure that that's under control. So, for example, when we decide between the aggressive prior and also the conservative one, we can see that the aggressive one tends to have a lot higher type 1 error inflation. So we want to calibrate against a strict control of type 1 error, considering all the scenarios that we investigated. Definitely that has the disadvantage. So in our case, it was quite clear that the conservative one would be the ideal choice in our scenario. I think there are many other things. For example, thinking about the different assumptions, the variations, or the sensitivity analysis that goes about understanding violations, right? Like a measure confounder and what's the impact on that, and whether you actually know enough information about the different prognostic factors in your study. So that's also important. I think the advantage of what we had was we have also some older studies that we can learn from understanding in this patient population what are the important confounders, and then what kind of information you have in regards to that, and if we control for that, assuming that, for example, I think in our setting, which I didn't cover today, we actually simulated taking out some of the known confounders and then see what was the impact on the type 1 error control and also power. And we actually see not so much, because I think we have the very good confounders all measured, yeah. Thank you.
Yeah, thanks for that presentation. You've got me thinking. There's many phase 3 programs done in drug development where a company will do 2 identical phase 3 trials, pretty much contemporaneous in time. In those trials, let's just say each trial has 1,000 patients. You put 500 on drug and 500 on placebo, and you've got 2 of those. Why can't we put 500 on drug and 250 on placebo in study A, and 500 on drug and 250 on placebo in study B, and borrow the placebo information across? Should be very few questions about exchangeability, or you could apply dynamic borrowing, but I would suspect you could borrow quite a bit of information, given it's an identical protocol done contemporaneously in time. And as long as your investigators were scattered kind of evenly between Europe, US, or whatever, you could have even geographic balance, if you would, between the 2 studies, and we could cut down on the exposure of 500 patients, placebo patients, and reduce time and cost. So is that an extension of what you're presenting here, or is that something that we should be thinking about routinely in drug development? And John, what would the FDA think about that idea?
So obviously I can't answer for the FDA, but I will say...
It's a review question, right?
Exactly. It's totality of the evidence. But it does seem like low-hanging fruit, and it's also the kind of thing that is not dissimilar to a master protocol. And so we do have sort of processes for thinking about those and reviewing them.
Yeah, so I think I agree with what John mentioned, and especially for non-oncology setting, like running 2 phase 3 is very common. And so actually, as far as I know, there are some examples of that thinking behind the scenes. I am not directly involved, but the methodology could be very closely related to the master protocol and similar. So certainly, I think related methodology can be used, but also I'm sure that you need to make sure that whether there's any issues that may come up, right, with such a design, yeah.
Yeah, I would just say one of the criticisms of some Bayesian and borrowing and all that as well, you still got to have enough patients exposed to your drug for safety and all that other kind of stuff. In the scenario that I mentioned or we've discussed here, you still are exposing the same number of patients to your experimental treatment, and so you're still accumulating sufficient safety data. And anyway, John, I liked your phrase about some low-hanging fruit, or consider it as a master protocol for your phase 3 program. Seems like it's eminently doable without huge leaps of assumptions or social or cultural or scientific barriers to overcome.
Dr. Lewis.
just want to point out something which you already know, which is when you share that control data, you're introducing a correlation in the results between the two trials. So you lose the independence of the two trials. So one has to think very carefully about whether you care about that for the particular development program.
I mean, I was even thinking, it doesn't even have to be Bayesian, right? It's like a multiple comparisons problem where you're comparing back to the same control group, and you could adjust for those correlations. Of course, there's the Bayesian. I don't know which one would work out to be better in terms of operating characteristics, but I suspect maybe some dynamic borrowing might be a reasonable approach, taking into account the correlation between the placebo groups. Anyway, just a thought.
I mean, it also raises the question of why not one larger trial? And I know there's an answer to that question, but thinking about what we get specifically from independent or quasi-independent replication versus more precise estimates from one trial, it's an interesting conversation, I think. Dr. Lee.
Yeah, following the discussion, I think we all like borrowing, right? Particularly if it's your money, right? And how to borrow it properly and really efficiently and get the more accurate decision. Yeah, it's what we are interested in. So in this case, Herb, as you mentioned, that R-CHOP has been used in the large B-cell lymphoma for over 20 years. There are ample of data, right? And the treatment is very standard. So you talk about hybrid control, which makes sense. So question one is that, again, how much, you have tons of data, right? So how much to borrow in this case, the hybrid control? I see many of the designs say that, we want to borrow up to the number of experimental arm, right? But rule of thumb. Or maybe, you can think about a factor of that, right? But in this case, there are so many data, and I argue that, nowadays with the very good electronic medical record and real evidence, this kind of getting the quality getting better and better. So one may think about the synthetic control, right? You don't even need a control group, right? So again, that I'd like to know your thought, and also John, FDA's thought on that.
Yeah, so I think certainly the real-world data setting can also contribute to the external control trials. So I think there are like two instances. If you can borrow from a concurrent ongoing one, and that already kind of is sufficient for the purpose of shortening time to read out for the overall survival when you do the primary PFS analysis, I think that's an ideal scenario you have, because I think even with very high quality data coming from real-world data setting, you may have other additional biases because subjects can be enrolled differently in trials versus more of the observational database. So there will be a high instance of like other biases that could be involved, but just need to make sure that like whether I think going back to one slide that we have is whether you have compelling effect size. I think if you have more compelling effect size, then you have more wiggle room, right, to have some of the risk, right, from the biases. But if it's more modest, I think it's safer to have a cleaner and higher quality data. So I think it really depends on the scenario. It's really case by case. I think in this particular setting, you have very ideal situation where you have a concurrent one. But in the case maybe you don't have one, it's because the concurrent one also is blinded, right? So I think that's the advantage too. The FDA also likes. But in scenarios where you don't, then I think you have to think about other options. With higher quality real-world data, this may not be a big issue, but you have to simulate, right? Also understand the scenarios where you have any other issues that may come up. Yeah.
I won't answer it in detail. First of all, because we're mostly here to listen to you all and draw our conclusions afterward. But I agree strongly with what Herb said, especially in terms of when you have a large treatment effect size, a lot of things are possible that may be less acceptable in a more marginal case. We see that in a lot of areas. And we are 5 minutes over for lunch, so we'll break for lunch. For folks who are here, there's lunch available for purchase right outside to the right. There's also some drinks behind you on the tables. Feel free to help yourselves. And we will resume at 12:30.
Thank you.
And going to proceed with the panel discussion part of the day. This will be the next 2 hours, and then we'll have another break, and then we'll have audience Q&A. So starting with the panel questions, I wanted to start, before we get into the sort of pre-set discussion questions, with a couple questions we've received online that I thought might be informative about the talks we heard. So there was a question for Dr. Lewis. How are futility analyses built into the simulations for type 1 error in power? Are the thresholds of 0.975 and 0.983 changed in the presence of futility analysis?
It's a great question. So the general principle, which I think many people are well aware of, is that the futility rule decreases the observed type 1 error if and only if the futility rule is followed. And so as a general rule, when we're simulating trials, we control type 1 error without the futility rule, and then we add the futility rule, which results in better control of type 1 error and sometimes a very small loss, typically a few percent in power under the alternative scenarios. Of course, the gain is that if the trial is trending towards a negative result, you can get out of it quicker, save resources, and save participants from avoidable risk. I think the type 1 error control, as I recall, is controlled without the futility rule. And if one was relying on your futility rule to maintain type 1 error control, then it must be absolutely clear that it is a binding futility rule and that there is no option for the trial continuing in sort of what we sometimes informally call blowing through the futility rule, because then you've lost your type 1 error control. I'm well aware that this is an area of some controversy, and I think the important thing is that those designing the trial and those who will ultimately be involved in reviewing the results and deciding its clinical impact have a clear understanding of the precise assumptions that underlie the calculation of the type 1 error control, and then are able to verify that the trial was conducted in a way that was consistent with those simulations and therefore has the desired operating characteristics.
Thanks, Roger. And we also had an online question for Herb. Does the hybrid approach require access to patient-level data from external controls, or could it be done with summary-level data?
Yeah, thank you for the question. So I guess in our case, for the analysis, we definitely need individual patient-level data. Because we are looking at the similarity between internal and external control, and then using it as a way to understand whether we should borrow more or less. But I think there are other approaches probably that can use summary level approaches, but that's not the case for our CID.